Wildfires have been filling the headlines all summer, and thanks to researchers working with the Environmental Molecular Sciences Laboratory, science could be moving closer to predicting these events and learning how they're changing the world around us. I'm Dawn Stringer with Bonding Over Science. Join me in learning more about how events like wildfires could be altering soil microbiomes. We all learn from a young age how to prevent wildfires from Smokey the Bear. But there's many other environmental factors that can contribute to these events that are deeper than we can see. Today's guest explains how EMSL is helping researchers like him understand these factors as they change with an evolving environment.
My name is Steve Allison, and I'm a professor of ecology and evolutionary biology, as well as earth system science at the University of California, Irvine. And my research studies the role of microbes in ecosystems and how those microbes respond to environmental changes like climate change and drought, and what the consequences of those changes would then be for our climate, our atmosphere, our soils, and ecosystems.
How did you get interested in that topic?
I guess I have to go back to probably my high school days or even childhood. I was interested in ecology from a fairly young age. My parents would take us out to various places during the summers. I lived in eastern Pennsylvania, and there wasn't a whole lot to do in the summers, honestly. So fortunately, my parents would take us on trips, me and my sister, through like the eastern United States at first, and then we would make trips out to Yellowstone or California, the West Coast. We went to Alaska one year, so that was pretty amazing. So I was always interested in nature, kind of being outdoors and seeing some of these iconic ecosystems. So then when I got to high school, I had a chance to take an AP biology class where we actually went to the Virgin Islands to study the recovery of corals and marine organisms from a hurricane that had come through a couple years ago. So that was a really fun experience and got me interested in biology. So I went to college at Penn State and studied biology and minored in chemistry there. So I kind of started broadening out from the biology focus to include some chemical interests, which I think is showing up in my collaboration with EMSL today. After that, decided I got into research as an undergraduate, studying plant-insect interactions, and then I wanted to go bigger and study the role of microbes in those ecosystems. So that was the topic of my dissertation, and that ultimately opened doors because at that time we knew that microbes were important, but we didn't have the tools yet, the sequencing and omics and other advanced molecular tools that we have now. And so that was all developing at the time. And so it's been nice over the course of my research career to be able to use those tools and approaches to understand and probe microbiomes in a way that we could not do, say, 20 years ago.
Can you explain, I read a little bit about your research, and wildfires they're included in there. What role do they play in your research in the environment, climate change, things like that?
Yeah, of course, there are a lot of impacts we expect to see from climate change. It's not just, warming of average temperatures, but we expect to see changes in rainfall and snowpack, temperature extremes, wind patterns, and all that. So here in Southern California, much of California, or even the Western United States, we're expecting a lot of these factors to develop together. And, we may have drier conditions. We may have more sources of fuel if they're invasive species, and we may have, more sources of ignition for fires, more interactions between people on the ground and the impacts of climate change. So that's what we're seeing here. And in addition to the drought conditions that we saw basically up until this year, where it got very wet in California, conditions were dry and we were contending with drought, but then also the wildfire risks. So we're seeing increases in precipitation extremes, but we're also seeing increased frequencies of wildfire. And again, that's partly due to the climate conditions, but also largely due to the fact that here in Southern California, there are 20 million people living in the area, and there are a lot more sources of ignition. So basically anywhere, anytime you have the potential for a wildfire, where the conditions are really dry, humidity is low, the winds are strong, there's almost always an ignition source around. So we're kind of just holding our breath, waiting to see when that's going to happen. And that's, that is what happens increasingly often as we see these large fires that may, burn up right to the edge of a city. And they have impacted our research here in Irvine. In 2007, there was a big fire, and in 2020, another fire that affected our research site.
So how important is it to study these wildfire trends and the impact it has on the earth beneath our feet, the microbes?
Wildfire has a big impact on all components of the environment. So of course it burns the plants, they're what fuel the fire, but it can also affect the surface soils and even deeper into the soil, depending on how strong that fire is and how high the temperatures get. So here in California, a lot of our ecosystems are adapted to fire. Fire is a natural part of the ecosystem, but we're seeing much more frequent fires, which is not natural, and that may have different consequences. But we are very interested in how the fires affect the microbes that live in the soil that provide these really important services and benefits to ecosystems, including plants, people, and the other inhabitants there.
What's a missing piece that with your research you're hoping to find between the genomic data and the microbial metabolism?
Well, there are many steps between the microbes and the other living components of our environment and the climate or the benefits that we see from our natural environments. So there's beautiful landscapes that we may go hiking in or enjoy, and that are storing carbon and probably helping us avoid more dangerous climate change. So we're really interested in knowing what are all the links between, say an individual population of bacteria and those broader important parts of the landscape that we care about. Not that I don't care about microbes, and we do care about microbes for the sake of the science and fundamental understanding. But I think if we want to connect, the microbiomes that are present in our environment with the factors that are really important to society, then we have to put this puzzle together. And so that's really what we're focusing on here, and especially how that puzzle is going to be scattered or affected by wildfire and other aspects of climate change. So, the basic principles of biology are that you start with genes, our genetic material that's encoded in our DNA, and that's the case for bacteria and fungi and the microbes that live in the environment as well. So that's the blueprint for what they can do. And then the next level is to say, well, what are they actually doing at a given time? And so for that, we can look at the expression of DNA. So we look at what genes are transcribed, what are showing up in the environment, out of that blueprint, what's actually being built. And then the part that's built are the proteins and the metabolites. So the chemical compounds that are produced by microbes or our own bodies. We can look at metabolites in the human blood. And so microbes don't have blood, but they do nonetheless have pathways, and they make metabolites. And those are really important for how they interact with each other, how they interact with plants or plant roots, and the impacts that they have on the soil and the environment. So then you have all these microbes, there's trillions of microbes, in a given patch of soil, for example, and they're all interacting and producing these metabolites. And so the next thing to do is to say, well, how does this all come together? And what does that mean for the plants in the system? What does that mean for how the soil and the plants might recover from wildfire? Is there a role in the prevalence of pathogens or the growth of the plants that we can attribute to this pathway from genes to the transcription to proteins and metabolites? So again, there's a lot of steps in that piece, which is why biology is really interesting to me. But we really need to probe each of those pieces very carefully and with the sophisticated instruments that we have at a facility like EMSL in order to really see what's going on in that puzzle.
Which instruments at EMSL are helping you answer some of these questions?
Yeah, EMSL has a really nice array of sophisticated instruments, and there's some that I'm not even touching because they have such a wide array of possibilities. But the kind of workhorse basic instrument that we're using is liquid chromatography, mass spectrometry, so LC-MS. And basically that is similar than it sounds. You take a sample, so some sample that contains the microbiomes, for example, that we want to study, or the plant material that we're interested in, maybe that burned up in the fire, and you mix it with some liquid, some solvents, some water, and extract out those metabolites. And then you can separate them out. So there's a separation phase, that's the chromatography phase. And then there's the mass spectrometry phase, which takes the separated metabolites and identifies them using like a chemical fingerprinting approach. So that's one of the techniques that we're using. And it's nice because we can kind of just look at what's there. We don't have to necessarily know what we're looking for, and it'll give us an indicator, like a list of the metabolites that are in the soil or in the plant material or in the microbes. And then we're also using another technique to look at the quantities of these chemicals. So we kind of get a list of what's there, but if we want to know exactly how much, then we may look at this NMR technique, nuclear magnetic resonance. And that's just a way to kind of focus in on certain chemical compounds and quantify, measure how much there is in a given sample. We're also looking at the structure of the soil. So the fire came through, and we're interested in the physical structure, like what does the soil look like from a microbial perspective. So that requires imaging in 3D, and we're using X-ray computed tomography to do that. And that'll give us an image of what the soil actually looks like, and if it's been affected by the wildfire. So I think those are the main techniques that we're using. And then, of course, there's a lot of analysis that has to happen after you do the measurements, and EMSL is helping us with that as well.
When all is said and done, what's your overall goal with understanding all of the aspects of this project?
Well, my goal is that ultimately we could actually predict what will happen in an ecosystem, right? We really want to know what the future holds in terms of climate change and wildfire. How bad is this going to be, or are there ways or opportunities that we can make it better, ways that we can prevent the worst from happening? So what I'd like to do is to take all the data on the genes, the blueprints that we have, and then the expression of those genes, as well as these metabolite data that we're getting from EMSL. And then we have a mathematical model, and we can take all that information. We have a team of postdoctoral scholars who are figuring out how to use that information and put it into the mathematical model. And then the mathematical model can make predictions about how the microbes are interacting and what that means for soil carbon storage, for example. Are these soils going to continue to store carbon, which is going to help us with the climate problem? Are they going to continue to cycle nutrients, which we need for plant growth and sustaining plants that are experiencing drought or climate change? And so that's the goal, is that we can make predictions, that we use our understanding to make predictions and know what's going to happen, and that we can do that at larger scales. So, microbes are tiny, we can't see them, they're microscopic, but they're abundant and they're really important. And we have to figure out how to take our knowledge of their genes and their physiology and their metabolism, and then use that to understand a whole ecosystem. So we can also use our mathematical models for that.
Can you explain how you heard about EMSL?
I've known about EMSL for a while. So one of my first grants that I received as a professor was in 2010 from the Department of Energy Genomic Sciences program. When you're fortunate enough to get one of these grants, you are able to go to the program PI meeting, principal investigator meeting. And so that's an opportunity for all of the people who are funded by that program to come together, usually outside Washington, D.C., and to meet each other and talk about the challenges and opportunities of their projects. And they also always have a session during that meeting on the capabilities of the program. And EMSL is always featured as one of those capabilities, right? Because like I said before, you have all of these instruments that, can really probe the chemistry and the biology and the interactions between microbes and their environment. And so that would appear every year. I would go to this meeting. And so I knew about the programs and opportunities and instruments at EMSL. So that's where I found out about it. And it was nice that we've finally had the opportunity to actually take advantage of those instruments.
Those are all the questions I had for you today. Is there anything you'd like to talk about that we haven't touched on yet?
Well, I would say just that, this is a long-term experiment, and we've had our trials and tribulations with the weather and the fires and, the disturbance in our research site. So I think this project comes at a good time, though, because we were actually able to see, this system go through a fire and collect samples before and after, and then have the opportunity to probe those samples in such great detail with these sophisticated molecular and chemical techniques. And so I don't know when we'll ever get to have that opportunity again. And it's been really nice that, they happen to align. I mean, of course, we weren't happy when our field site burned down in 2020 during that fire, but we were positioned, because we've been working there, to take advantage of that as an opportunity for learning and new knowledge about how the system changes with fire, which again is an increasingly frequent occurrence in our system. So I guess we're trying to make lemonade out of lemons, and it's been really nice that we have a background of collaborators and opportunities through EMSL and through our funding agencies to keep that work going and to see how it pans out over the years.
Awesome. Well, we're excited to see where your research goes in the future.
Awesome. Yes, I am too. You never know what's going to happen.
Thank you for listening to Bonding Over Science. I'm Dawn Stringer for the Environmental Molecular Sciences Laboratory. We don't have time to cover it all, so don't forget to check out emsl.pnnl.gov for a full article on this topic featuring who I spoke with today. And don't forget to follow us on all social media platforms for the latest and greatest news coming from EMSL. EMSL is a Department of Energy Office of Science national user facility that accelerates scientific discovery and pioneers new capabilities to understand biological and environmental processes across temporal and spatial scales. EMSL leads the scientific community toward a predictive understanding of complex biological and environmental systems to enable sustainable solutions to the nation's energy and environmental challenges. If you're interested in working with EMSL, learn more at emsl.pnnl.gov. That's emsl.pnnl.gov.